Okay, so we left off talking about these sensory receptors last time and the different classifications of those. And so we'll just talk about those for a little bit more, and then we'll dive into the reflexes, okay? So the temperature ones here, the ones that detect temperature, are called thermoreceptors. There's different ones for cold than there are for warm, okay? And one thing that's important about these is they detect changes in temperature, okay? So changes in temperature only, okay? So cold ones detect cooling, right? So when temperature is declining, that's what they detect in that range, okay? And then the warm ones detect warming or increasing in temperature in that range, okay? And that's just the basics of that with the thermoreceptors. Have those all over your body, mostly throughout your skin, all over the place. Okay, pain receptors, I mentioned the name of these last time, nociceptors, okay? There's lots of different types of nociceptors, okay? So these could either be chemoreceptors, right? So they're detecting chemicals that could be potentially harmful for you, so toxins. One good example is histamine. It's a chemical released by inflammatory cells as part of the inflammatory process, partially what causes pain when you have inflammation, okay? Same thing with cells. When cells are destroyed, when you have an injury, they release a bunch of substances. Some of those substances directly activate pain receptors, okay? We have hot pain receptors, cold pain receptors, pinch or kind of intense pressure pain receptors, okay? So all detected by different types of receptors there. Okay. All right, all of this sensory information is going to go to the brain, obviously, and it's going to get processed there, right? So processed in what's called the somatosensory cortex. And where is that in the brain? This is what we talked about this last semester, the different parts of the brain. The parietal lobe is where we find the sensory cortex, okay? And we can see that here, right? So it's kind of the very front part of the parietal lobe before we get to the frontal lobe, whereas the primary motor cortex is right here. That's part of the frontal lobe. Okay, we have this situation where the brain is set up in such a way, oops, didn't mean to get that thing, set up in such a way where we're going from the middle of the brain, so the medial side, right, all the way to the lateral side over here. All of the sensory receptors for certain regions of the body are together, right? So all the ones for the feet are right about there, right? All the ones for your face are right in this region, right? And so the more sensitive a certain area is to touch, then the more receptors it has dedicated to that area, which means that part of the brain is bigger because all it is really neurons in this area. That's where the cell bodies of those neurons are, and then they send their axons out to that part of the body. So the larger the region is in this part of the brain, the more sensitive that part of the body is to touch. Okay, and this thing, there's something called the sensory homunculus. Okay, you can kind of see I have the word kind of halfway covered up there, but it's just missing the HO part, but homunculus, okay? And really you can take this person here and put it all into an actual person that's like standing up, and all the different body parts are different sizes depending on how much sensory information you have for each. And you'll see that in your book. It's called the sensory homunculus. So it has these huge lips, right? Tiny other parts of the body that, and really the size of the body part is just reflective of how many sensory receptors are found for that part of the body. Okay, questions on that? Okay, all right, so let's talk about reflexes. So reflexes are these automatic reactions, as you're familiar with, that occur without conscious thought, which really means without the brain. Okay, so the brain does not have to be involved, but it can be. Okay, so reflexes occur they're called spinal reflexes because something comes from the periphery to the spinal cord, and the reactions happen right away, right back from the spinal cord without using the brain. Okay, that's why they're called spinal reflexes, but your brain can modify that if it wants to. Okay, if your brain doesn't sense that there's truly a threat there, then the brain won't let the reflex happen, right? Like when your friend comes up behind you when you're studying, taps you on the shoulder, that could create a startle response, right? If you're not ready for it, okay? But you could start to have that response, but then you could modify it, right? You could dampen it down because you automatically realize there's no true threat to you, right? So that's just one example of that. Okay, you'll work with some of that stuff in a lab coming up, not this week, but the next week. Okay, and so all reflexes involve the same basic things. They all involve a receptor, a sensory neuron, place where that information gets integrated in the spinal cord, a motor neuron, and then an effect, right? Where it goes out to a certain muscle and has an effect. Okay, so we're going to look at a couple of those right here. Okay, two special reflexes driven by two special receptors. Okay, the first one's called the stretch reflex, and the receptor for that is called the muscle spindle, and it's shown right here. So we have little tiny muscle spindles embedded in all of our skeletal muscle throughout the body, and they look something like this, right? So this kind of interesting type of skeletal muscle, but wrapped with nerve fibers. And what it does is it tells the body, or it tells the brain at all times, what is the length and the position of every single muscle in the body, right? And so this is pretty incredible, the amount of information that's coming into your brain from these little guys all the time, right? And this is really the only way that we can maintain posture, the way we can stay upright, the way that we don't fall over, right? Is because your brain knows exactly how long and how much and the position of every muscle in your body at all times. All this information is going into the cerebellum processes that. If all of a sudden I start leaning over to my side, right, then what happens is I start stretching out the muscle spindles on the opposite side of my body, the side I'm leaning away from, and the cerebellum senses that because of these muscle spindles, and it tightens up that side a little bit so I don't fall over, right? And that's exactly how we maintain posture, right? So that's very, very important. So that's the muscle spindle, and they're involved in the stretch reflex, which we'll go through in 1 second. And then secondly, there's another one called the Golgi tendon organ, also known as the GTO. And these are embedded in all of our tendons, okay? And these ones are very important for what's called the deep tendon reflex. And we'll talk about that one here in 1 sec as well. Okay, so let's walk through the first one, the stretch reflex. Okay, so the stretch reflex, all reflexes have a purpose, okay? And so the purpose of this one is to prevent overstretch of a muscle. Let's not let a muscle get too stretched out because that could damage it, okay? The receptor for that, as I just said, is the muscle spindle. What is the stimulus that the muscle spindle is detecting? It's detecting stretch of a muscle, okay? And stretch includes length and position of a muscle. Okay, so it's not just that it's getting pulled, it actually knows some sensations being sent to the brain that kind of gives the brain an idea, not just that it's being pulled, but how long it is, because it can be pulled from a shortened position, or it could be pulled really fast from a lengthened position, and the brain knows the difference between those two stimuli with this. Okay, so the pathway here is shown, and I'm just gonna, these steps are important, okay? So you definitely wanna know these steps and memorize these steps, but I'm gonna show how they play out right here on this image. Okay, so this is the same reflex that the doctor does to you when they test your reflexes, right? They take that reflex hammer and they hit it against your patellar tendon, right? So that's the stimulus. It hits there, hits your patellar tendon. Your patellar tendon is connected to your quads, right? Your quadriceps. Okay, so by pulling on that tendon a little bit, we create a little bit of stretch right there in the quadriceps, right? So number one there is we activate the muscle spindle, right? So we stretch quads, activate the muscle spindle in the quads. Okay, so that's number one. And then number two, that's going to create an action potential, AP, on an afferent neuron. Right, so there's an afferent neuron, a neuron heading back towards the spinal cord. Is that coming from mine? No, it is. Here, let me do this. That's what I got to do. I'll forget. Okay. All right. Yeah. So that sends an action potential back on this afferent neuron. So that's number two. That's going back to the spinal cord. Right. And you guys remember all this setup of the spinal cord from last semester, right? We go through this guy right there. right? That's where the cell bodies of those sensory neurons are. Remember that? That's the dorsal root ganglion, right? We're going back into the root here, and now we're into the spinal cord, okay? So that takes us that takes us through 2, and then 3A is shown in this pink one. We see a plus sign right there. We see this sensory neuron going right to that pink one, okay? And so what's that going to do? That is going to cause contraction. So we activate a motor neuron on the quads, then we contract quads. Okay, and this makes sense, right? If we're trying to not let the muscle stretch out too much, the thing that would make the most sense would be to actively shorten it, right? Which is what contraction does, right? So we're trying to go against that stimulus. That stimulus was stretching, so we want to go against that stimulus of stretch and contract instead. So the quads contract, and that's exactly why your knee kicks out when you do this, right? Okay, so that's action number 1 in 3A, but then we see 3B right here too. We see 3B, it has that little green neuron. That's called an interneuron, right? That's just neuron to neuron communication. And so we're going to activate inhibitory interneuron. We're going to activate that inhibitory interneuron, and then we're going to inhibit the antagonist muscle. Okay, everybody in the chat right now, throw in the name of the antagonist muscle group of the quads. What would be the antagonist of the quads? Throw it in the chat. Okay, we got a couple. Keep going. Throw it in. There we go. Good. Hammies, hamstring. I like whoever said hammies. Good. Perfect. Yeah. So you guys know that. Okay. So let's go into the hamstrings right there. Okay. So we inhibit those, right? Why would we inhibit those? Because we don't want to. If we contract the hamstrings, then we're going to stretch the quads more, right? That would be the opposite of what we want to do. We don't want the quads to stretch, right? Because we're going against the stretch. Okay. So there's 2 actions to all these reflexes. Okay. First action is to contract the one being stretched. Second action is to inhibit the antagonist, which could cause more stretch if we let it. Okay. So that's that. Okay. And that's all explained here in these steps as well. Okay. But I just kind of wrote it out in the picture here. All right. So definitely a process you want to memorize and understand. Okay. So questions on that one? Okay. All right. So we got some other reflexes, obviously many other reflexes. The deep tendon reflex is the one that uses the Golgi tendon organ, or the GTO. Okay. Its purpose is to protect against overcontraction. Okay. So this keeps us from tearing our muscles, because our muscles are actually way stronger than we ever let them actually contract. Okay. If we let our muscles fully contract, we can actually rip our tendons right off of our bones. We can actually break our own bones with our muscles because they're so strong. But instead, we have the nervous system involved with the Golgi tendon organ. And the Golgi tendon organ senses when there's too much tension on a muscle, and it forces it to relax. Okay. So the stimulus is tension in the tendon. So just how hard is it being pulled on both sides? And that causes the exact opposite of what the last one just causes. It causes relaxation of the contracting muscle. Okay, so that one that's contracting, it causes it to relax, and then it's going to cause the other one to contract, the antagonist. Okay, so it's just pretty much the opposite effect of the muscle spindle. Okay, and then the flexor withdrawal reflex, we're all used to this one. If we touch something hot or we step on something sharp, the receptor is a pain receptor. This causes us to move away from the dangerous stimulus. And it's all of our flexor muscles at the same time, right? So we touch something hot, what happens? Our finger flexes, our wrist flexes, our elbow flexes, our shoulder, everything is flexing at the same time. And that's why it's called the flexor withdrawal response. Okay, any questions on those?
I just had a question. I was wondering if there's any condition that how do I phrase this? Is there a condition that the person doesn't have these reflexes, I guess?
Sure, not that doesn't have them, but there's definitely conditions where the reflexes are dampened or over, or you can have hyperreflexivity, where these are kind of too strong, or you can have hyporeflexivity where these aren't strong enough. even just anything that messes with your nervous system, like, so for example, diabetes damages your neurons. And so that dampens reflexes majorly because your neurons are not able to communicate as rapidly anymore. Having alcohol on board dampens reflexes, right? Because that messes with it actually would increase the reflexes because your brain is too slow to involve itself in the reflex and doesn't stop the reflex from happening. So there's lots of different things, but definitely many conditions affect reflexes, really anything that affects your neurons. Yeah. Okay. Yeah, so let's go ahead and do this one here on the app. So go ahead and open that up. And yeah, these are choose all. So I'm going to do multiple answer. Okay, go ahead and think through this one and just put in all the ones that you think are correct. So I think with this one's a little bit different. You just put in the multiple letters there in a row and then hit submit or send or something like that. Okay, give you about 20 more seconds to respond. All right, shutting off in 5 seconds. Send something in if you haven't yet. All right, I'm gonna go ahead and shut that off. Okay, good. So yeah, so yeah, did it let you put in 2? It did. This is a different output than I was used to, but yeah, okay. There we go. That's the one I was used to seeing. Okay, yeah. So most of you are putting in B and C. All right. So when we look at this, so muscle spindles sense stretch and Golgi tendon organ sense tension. Okay. And again, the difference between those two, stretch has a component of length to it. Okay. So it's not just pulling on both sides. It's the pulling, but then also a component of how long is the muscle at the same time. Okay. Whereas the Golgi tendon organ, that's really just how hard are we pulling on both sides of it. Okay, so that one's right. And then C, stimulation of the muscle spindle causes contraction of the agonist muscle. That's the one being stretched. Okay, and inhibition of the antagonist muscle. In our example, that was the hamstrings. So that's right. The stimulation of the Golgi tendon organ causes contraction of the agonist, the contracting muscle. That would be the opposite. The Golgi tendon organ is protecting us against too much contraction. So it would actually cause inhibition or relaxation of the one that's contracting. Okay, so any questions on that? Okay. All right, and this is kind of what I just talked about in response to Rachel's question, hyperreflexivity versus hyporeflexivity. So anything that kind of gets in the way of your brain dampening a reflex will cause hyperreflexivity. But then anything that gets in the way of the neurons passing the signals back and forth will typically cause hypo or low reflexes. Okay, and that is it for that. And we're going to move on to the eye here. Okay, so this will just be your second set of notes there, and let's jump right into this. So kind of a beautiful image of the human eye there. The iris always looks somewhat magical in a way when you look at it closely, right? It just kind of has this ethereal look to it. Yeah, so the human eye contains 70% of all sensory receptors in the body, right? So of all of our sensory receptors throughout every square millimeter of our body, 70% of them are right here in our eyeballs. Okay, that's 126 million sensory receptors in our eyes alone. Okay, almost half of the cerebral cortex, which is the big top part of our brain, is involved in vision in some way. Okay, so it's no overestimation to say that humans are vision dependent and vision dominant as far as their senses go. Okay, very dominant sense in humans. Not true for many other animals, right? But in humans, vision is the primary sense for us. Okay, so today will be mostly anatomy of the eye, and then Friday's lecture we'll get into the physiology and how all these things work together. Okay, so first, just some of the accessory structures and the things we find around the eyeball. Okay, so eyebrows, right? These give us some shade. These help us catch sweat when it's falling down our foreheads. Okay, eyelids. There's a different type of reflex, but reflexive blinking every 3 to 7 seconds is what happens in our eyes. And the purpose of that is to spread the lubricants that are being released by glands in the superior part of the eye. Okay, so to spread those lubricants around. Okay, also the reflexive part of blinking, when something goes towards my eye, my eyes will blink, right? Or my eyelids will close. That's obviously to protect you from something entering your eye. Okay, so the glands that release the lubricating fluid are called tarsal glands. Okay, they secrete this oily solution that lubricates the eyelid and allows the eye to move a little bit easier. Okay, good there, ready to go on? There we go. All right, so a couple other accessory structures here. So the conjunctiva, this is the mucous membrane that lines the inside of the eyelids and the white portion of the eyeball. Okay, so all the white part of your eye, so not around the iris, but everywhere else, not around the iris and the pupil, but all the white part. And then on the inside of your eyelids, you have this mucous membrane. Okay, and this again is helping to keep the eyeball moist, helping to keep the movement of the eyelids fluid, and we can irritate this, and that's called conjunctivitis. Right? Itis means inflammation, so it's inflammation of the conjunctiva. If this irritation is caused by a bacteria or virus, then this is what's known as pink eye, like this guy has here. Right? Highly contagious if it's the viral type. And if it's a bacterial type, it's much easier to knock out with some antibiotic eye drops. But if you have the viral type, it can be pretty nasty. Put your hand up if you've ever had the viral type. It takes like a week or two to go away. Yeah, it's not fun. I had it on a Hawaii trip one time, and it was terrible. Yeah, it's like my eyes are just leaking like the whole time we were there. It's just all swollen and nasty. Yeah, and there's not really much you can do about the viral type. You got to let it run its course. So yeah, that's pink eye. Okay, so other accessory structures here. We have this thing called the lacrimal apparatus. Okay, and that includes the lacrimal gland up here in the upper lateral corner of your eye. Okay, and then that's going to release tears. Okay, going to go through here, and then if you remember last semester, one of the bones you learned was called the lacrimal bone. Right? Remember that little lacrimal bone that was right here and it had a hole in it. Right? That lacrimal bone actually had a hole in it. That was what made it a distinct bone to remember it. And it has a hole in it, so these tears can go through. And that connects to the nasolacrimal duct here, and then that connects right into your nose. Okay, so this is absolutely why when you're crying, you also get a runny nose, right? Because the excess tears going through your eye end up into your nose because of this system here to deal with the excess tears. Okay, so the function is there's mucus in here, which is for moisture and lubrication and protection. There's antibodies in here, so part of your immune system is in here dealing with any invaders that might be getting into your eye. There's also lysosomes, which are special proteins that destroy bacteria. Okay, and normal tears are stimulated by the facial nerve, cranial nerve 7. Okay, this just kind of walks you through the what I just kind of did there with the flow of the tears. Okay, so normally we have just kind of this constant steady flow of some fluid leaving our tarsal or leaving our lacrimal glands there. Okay, so a normal steady flow, but then obviously in times of emotional distress, we cry as well. And there's actually some physiological parts to this. The tears that come out during times of emotional stress actually contain the neurotransmitter, a group of neurotransmitters called enkephalins. And enkephalins actually cause you to relax. They produce feelings of peace, and they're kind of part of your feel-good neurotransmitter groups. And so we don't, normal tears don't contain that, but when you get that emotional crying, it actually contains enkephalins, and it actually makes you feel better. So just cry, guys. I know you're thinking not supposed to cry, but it's all right. You got to let it out sometimes. And girls. I wasn't just talking to the guys. I know some girls think they can't cry either. Okay, any questions on that? All right. Oh yeah, sorry, I forgot that one part there. So if you ever have a blockage of the nasolacrimal duct, which would be this part here, right? Then you get this mucus buildup kind of in the corner of your eye, and your tears don't have anywhere to go because of that, and that causes that mucus buildup. Really common in newborn babies, and they kind of get this crusty stuff all in their eye. And literally, you just have to kind of take your finger and kind of smash it around in the corner. It helps break it up, and then their tears can flow more freely, and you don't get that mucus buildup. So it's similar to what you have when you wake up with like sleep in your eyes. Yeah, the last box on the bottom, that was that one right there, the teary eyes, mucus buildup. Is that what you're asking about, Joanna? Yeah. Cool. All right, any other questions on that? Okay. All right, so just as a quick kind of recap here, this would be something I'll let you guys do on your own, but it would just be kind of a recap of what we just talked about, location and function of each of these accessory structures that we just talked about. Okay, so we're not going to do that right now. I'm going to move on, but I'll just be a way for you to kind of recap that. Okay, so let's start talking about the eyeball itself. Okay, and we've kind of got these three layers of the eye called tunics. Okay, so 3 tunics. And we'll start with the most exterior, or the most outer one first, which is called the fibrous tunic. Okay, and the fibrous tunic contains 2 different parts to it. It contains the sclera and the cornea. Okay, so the sclera is the white part of your eye, and everywhere that's not clear in the front and all the way around the back is part of the sclera. So this is very thick and tough, fibrous, gives the eyeball its shape, serves as an attachment point for all of the muscles that move the eye. Okay, and then the very front part is the cornea. Okay, and the cornea has some epithelial tissue on the front. Okay, and it contains these special proteins that are completely clear. All right, and they're called crystalline proteins. Structure shown there. Okay, and the function of the cornea is to start bending the light as it passes through the eyeball. Okay, so the light's going to pass through the cornea first. That's the first convex surface that it has to pass through, and that does a lot of the initial bending of the light to start getting it closer to being focused. But then the lens is going to help us out with the rest of that, but the cornea does the initial part of that. And so you have lots and lots of nerve endings in the cornea. And so for those of you who have ever scratched your cornea, you know what I mean by that. It's extremely painful. Okay, you can scratch the white part of your eye, the sclera, and it doesn't really hurt that much. But if you do get some scratches on the cornea, it's a very painful injury, and it can take a while to go away. Okay. All right. So that's the most exterior one. That's the fibrous tunic, and then we'll move into the next one, the middle portion, which is called the vascular tunic. And the vascular tunic contains multiple structures here. So the first part is called the choroid, and this is the back part of it, the posterior region, very blood vessel rich, okay, and contains pigment cells. And so the function of that is to, number one, the pigment cells help us to absorb light and minimize scattering of the light that comes in, okay, and you'll see this in lab when you dissect the eyeball coming up soon. Okay, you're going to dissect a cow eyeball, and you'll open it up and you'll see the choroid inside. It's a cow eyeball, so it looks a little bit different than ours, but so we get the light absorption, minimized scattering, but then blood supply being supplied to the rest of the eyeball through this region. Okay, this whole idea of a deer in headlights, right? When a deer gets in the road and looks up, and those bright headlights go right into its face, they don't have a very prominent choroid layer in their eye. They don't have that pigment-rich part. So when a bunch of light comes into the eyeball, it scatters all over the place. It pretty much knocks out all of their sensory receptors. So it literally blinds them for a few seconds there. And yeah, and so then they can't see, and they're kind of just stuck there, right? And so humans have a little bit less likely. That's less likely to happen in humans because of the fact that we have that light absorbing material inside of our eyes. Okay, another part of the vascular tunic is called the ciliary body. This is the very anterior part of this region. Okay, this is smooth muscle that's under the control of oculomotor nerve, cranial nerve 3. And this is important. This is a muscle that controls the shape of the lens. I'll talk about the details of how that works in a bit. That's the ciliary body. Okay, and then the ciliary processes are right there in that region as well. The function of those is to secrete the fluid that we find in the anterior part of the eye called aqueous humor. Okay, so aqueous humor is found in the anterior part of the eye in front of the lens, and it's very fluid-like. Okay, and that's secreted by the ciliary processes. Posterior part of the eye is filled with vitreous humor, which we'll see in a second. That's more jelly-like. The part in front of the lens is aqueous humor, which is much more liquid-like. All right, questions here?
If you get something in your eye and it's like really painful and your eye starts watering, what is really like being harmed or affected?
Most likely your cornea. Yeah.
Okay.
Yeah, because it doesn't actually get through, because the cornea is a solid structure, even though you can see through it. But if something gets stuck on your cornea and then you move it around with your eyelid, sometimes it can kind of like drag it over your cornea, and if it's sharp enough, it can scratch the cornea, and that's pretty painful. And then it'll just be kind of watering for a couple of days typically.
Mm-hmm.
Yeah.
Thank you.
Yeah, for sure. Other questions on that?
I had a quick question about like
Sure.
since we talked about a little bit on the pigment. So does that mean like brown eyes would be able to resist more to like say bright light, and like blue eyes would be more sensitive?
Good question. It's a little bit different than that. The pigment that we find in the iris is what gives our eyes their color, which is different than the pigment that we have in the choroid region, which is more like the posterior part of the eyeball, kind of all around the interior part of the eyeball. What you're seeing with the eye color is in the iris, which is more, doesn't necessarily have as much of a light absorbing function. It does do that, and darker eyes will absorb a little bit more light in the front, but that's not really the primary place that light needs to get absorbed. It's more once it gets into the eyeball. But yeah, same idea. Definitely those pigments are light absorbing proteins, and we have them in our iris as well as in the back of the eye too. Yeah. Okay. All right. So let's keep going with this, looking at the this middle region still known as the vascular tunic. So there's two different. There's the iris, as we were just talking about, and that's made up of two different muscles. Okay. So the pigment content, as we were just talking about with Hannah's question, determines the color of the eye. Okay, so the iris is one of the muscles, or it was two separate muscles inside of the inside of the eye, and there's two different muscles in there. We'll show them to you in 1 second, and they're colored because of the pigment inside of them. Okay, and their job is to determine how wide open that pupil is. Okay, really, and that helps us determine how much light can enter. So the pupil is literally just a hole, right? The pupil is not really a structure, it's just a hole, okay? That's the black part in the middle, and that's what lets the light in the eyeball. Okay, so let's look up here on the top part of this. When we see this diagram, you can see clearly there's two separate muscles, okay? So there's just one in the middle, it's a circular muscle. That one's called the pupillary dilator muscle, okay? And then there's this one on the outside, which the fiber's going in this direction more, which is called the pupillary sphincter muscle. Okay. And so the important part to understand here is the one that we're typically using is this one in the middle. Okay. The one in the middle, and that one is controlled by your parasympathetic nervous system. Okay. Yeah, so that one is typically going to be going to either when it contracts, it squeezes down and lets less light in, and when it relaxes, it opens up and lets more light in. Okay, so when we want to let less light in, we constrict the pupil by using our parasympathetic nervous system, using our oculomotor nerve. We cause that contraction of the pupillary sphincter muscle. Okay. And so we're going to do that in situations where we're close to an object. When we're really close to an object, we want to let less light in, and so our pupils are going to constrict. So that's when we use that for near vision. We'll get into that more for Friday's class. Okay, and I'm sorry guys, these are pointing to the wrong ones. The pupillary sphincter muscle is the one in the middle. Okay, and this one, pupillary dilator muscle, is the bigger one on the outside. Okay, I'm sorry, these little blue arrows are pointing to the opposite ones. Okay, so all sphincter muscles are circular muscles. Okay, and so that everybody got that? Sorry for that confusion. Those blue arrows are pointing to the opposite muscles. Questions on that to clarify that? Apologize for that mistake.
Does that mean you use the outer one more often?
No, what I was saying there was right. It's just that those two blue arrows were pointing to the wrong spots. Yeah, so you use that middle one most of the time when we're just under normal conditions. That's one's controlled by our parasympathetic nervous system. Okay, but then when we want to dilate our pupil, most of the time, if we're just normally in a situation where, let's say, an object is further away from us, that we need to use it, then what we do is we actually inactivate the oculomotor nerve, relax that sphincter muscle, and that causes dilation. And we do that for far vision when we want to let more light in. Okay, so that's all driven by that pupillary sphincter muscle, either contracting for near vision or relaxing for far away vision.
So is it more that the pupillator or the pupil dilation is like just what would happen naturally, but if you weren't constricting the constrictor muscle at all, like it would just, you'd relax and it would always be open like that?
Yeah, so yeah, exactly. Yep. So relaxing, and this is true for our skeletal muscle too. Relaxing a muscle a lot of times is just as functional as contracting a muscle, and it's definitely the case in this case. So when we relax that sphincter muscle in the middle, it opens up our pupil versus contracting it when it closes it down. So yeah, that's as you said, Caleb, the more natural state would be just relaxing it and letting it open up.
So the dilator muscle isn't really like innervated at all?
Well, no, so we'll get to that in one sec. So the dilator muscle is controlled by your sympathetic nervous system. I can't remember if I had something right there. Yeah, I didn't. So that's driven by your sympathetic nervous system. And what it does, you can see these little arrows, it actually just attaches, it's attached to the sphincter muscle in the middle. And so it literally just pulls open that sphincter muscle. Okay, and it'll do that under conditions of stress. So in the special senses lab that you guys will have in a couple of weeks, you're going to have your partner's going to pinch the back of your neck. I can't remember if we're actually going to have you guys touch each other this time around. Probably not. You might just pinch your own neck, but it doesn't work as well. But when that happens, if you're watching somebody's eye, their pupils will dilate for a second really quickly. Whenever there's pain or some threat to you, your pupils open up really quickly. Right? And the reason that makes sense is it's important to be able to see clearly, or when it's important to see clearly, your pupils will constrict so you can focus in on one thing. But if you're under attack, you don't really care if you can clearly see your attacker's face, right? Or you can clearly see what animal that is chasing you. You just want to see it, right? And so you want to open up your eyeball as wide as possible so you can have as big of a range of vision, right? And so that sympathetic activation of the dilator muscle just opens it up so you can see a lot. You can't see it very clearly, but you can see a lot, right? Whereas the other one is always working more just to kind of fine tune our vision under conditions of not being stressed. Okay, questions on that?
So does farsightedness or nearsightedness have anything to do with how like well, I guess, the pupillary sphincter muscle dilates or just
No,
functions?
it doesn't. That's a good question. We'll get to that for sure. But that really has more to do with where the eyeball ends up focusing the light, whether it's in front of the retina or behind it. And that more has to do with just the size of your eyeball, interestingly enough, if your eyeball's too long or too short. And so yeah, we'll get to that in a sec, though. Good question. Yeah.
And I have one more question, if that's
Go
okay.
ahead.
So, you know, like when you look at someone who's blind, their pupil might be a little more cloudy or catch light in a different way. What is the cause of that?
Yeah, that's a great question. I don't really know. I would think that the cloudiness might just have something to do with more with whatever pathology it is in their eye that's causing that. It might just be some degeneration of the retina that's not really being used. And so when you look into somebody's eye, you don't see the same color because the retina is not actually the same type of tissue. It's kind of degenerated. But then there's also, a lot of times when you look at somebody look at somebody's eyes who are blind, they're not necessarily just tracking the same, right? Because so there is some loss of the ability to track because they're not really needing to track because they're not looking at the same thing. So the brain doesn't control the eyes quite the same way. But yeah, my guess would be on the cloudiness that it's just the structures behind the cornea are actually just kind of deteriorated or have some kind of pathology in them. Yeah.
Okay, thank you.
Yeah, good questions. Any other questions on that? Okay. All right, so let's take a look at this one here. And I'd like you to answer these questions here. So circle one of each bold and underlined word, okay? Then send in both letters on your clickers, okay? So so under situations of stress, we dilate our pupils via contraction, relaxation of the pupillary dilator or sphincter muscle, okay? But under situations where we need to see something far away, we dilate our pupils via contraction or relaxation of the pupillary dilator or sphincter muscle, okay? So so go ahead, and so it's just the C or the C or the, I don't know why I had, so it's C would be the contraction part, right? D is dilator, right? C is contraction, S is sphincter. Does that make sense? I was having to remember how I set that up again. Yeah, so it's just whatever the first letter of the word is, right, for those. Okay, so take a second and I'll open this up. And again, so you're just going to send A through D or A through D for each of them. Okay, so you'll send it twice. So you could send AB or CC or whatever it is. All right, maybe about 10 more seconds. A lot of letters there. Could get a little confusing for sure. Okay, go ahead and send something in. If you're still not quite there, just send something in so you get the participation credit. Okay, so we're, it might be better to look at it this way. I don't like the new way this thing's showing it. It's not how it used to show it. It used to show them grouped together, but it's not showing it that way now. It makes it trickier for me to interpret here. Well, Yeah, so it looks like B and D are kind of the most prominent ones there. Okay, but it's hard to tell which order they're in with this new output that the software has given me. But yeah, so let's take a look at this. So under situation of stress, we dilate our pupils via. We were just talking about that. That's going to be that dilator muscle, right? But the question is, do we contract it to do that, or do we relax it? And in this case, that's going to be contracting that one, right? So that's going to be the contraction part and the dilator muscle, because we're talking about stress, we're talking about our sympathetic nervous system in that case. So that would be the CD, so that would be A. Okay, but under situations where we need to see something far away, not a stressful situation, we dilate our pupils via relaxation of the pupillary sphincter muscle. So that would be RS.
For the first one,
Mhm.
I thought you said like if we're being attacked, our pupils would get really big.
Mhm.
Wouldn't that be relaxing the muscle?
So we're talking about the other muscle, right? So
Oh,
yeah, that's where this is confusing. That's exactly why I added this question for this year. I hadn't had these in the slides previously. So sympathetic activation, which would mean stressful situation, we're going to dilate via contracting the pupillary dilator muscle. But if we're just trying to see something far away, we're not stressed out about it, we're going to not use that sympathetic nervous system anymore. We're still using the parasympathetic. So we're going to just relax the other one, that pupillary sphincter muscle. Okay. All right. So don't worry too much if you're getting confused by these letters down here. That's not the important part. Obviously, the important part is just the way knowing which one needs to contract and relax under different situations and what effect that has on the size of the pupil. Okay. Any other questions on that one?
I have a quick question.
Sure.
Does that mean the dilator is only used for in the sympathetic nervous system only?
Only, yep. It's
Okay.
controlled by sympathetic nerves, whereas the pupillary sphincter muscle is controlled by parasympathetic nerves, the cranial nerve 3. Right. So
Okay.
So they're totally separate in the way that they're activated. Different parts of our brain activate them.
Thank you.
Yeah, good question. Other questions on that?
Just to clarify, is it the pupillary sphincter muscle that's primarily used?
Yes, that's the primary one. Mm-hmm. Good question, Justin. Okay, anything else on those? All right, so what I will do is we're out of time for today. So what I will do is I will go ahead and I'm going to pause the recording here for a second. All right, guys, so we're going to go ahead and continue here with the rest of these notes. And so we just left off talking about the different tunics so far, and we've talked about the outer and the middle tunics. And so then, but this image here includes the inner tunic, which we haven't talked about. But I would encourage you to pause the video for a second and see if you can label these different structures in the eye, and then you can restart it and see how you did. Okay, and there's this question down here: which is the muscle that controls how much focusing happens to the light that comes in? Okay, so let's look at each of these. So A is that part of the external tunic called the cornea. B is the choroid layer. You can see it's nice and thick and has blood vessels and is pigmented. C is the retina, so the very internal tunic. We haven't talked about that one yet. D is the iris, where we talked about that. So that's going to be right here, controlling the size of the pupil. E is the ciliary body. We can see how it's right there. That's attached to these little ligaments, which are attached to the lens, and that's going to control the size of the lens and the focusing of the light. Okay, so which muscle is it that controls how much focusing happens, the light that comes in? That's the ciliary body. Okay, and then the sclera is that external white part of the eye, right? And then G, the big optic nerve coming out of the retina. Okay, so let's talk about the last tunic, which is the most inner layer, which is called the sensory tunic or the retina. Okay, there are two layers to it. There's a pigmented layer, and then there's a nervous layer. Okay, so the pigmented layer is right up against the choroid, and its function is to also help prevent the light scatter. And it also replaces damaged photoreceptor cells, because those photoreceptor cells have pigment in them as well. And so pigment being available right there next to the nervous part of the retina that has those photoreceptors is exactly what we want. So let's look back on that so we can see the retina here, right? So here's the choroid, here's the retina. You can't really see the two layers of the retina there, but the side that's closer to the choroid is the pigmented layer. And the side that's further out here is the nervous layer. Okay, so the nervous layer is the innermost portion, contains photoreceptor cells. We've got two main types, the rods and the cones. The rods are for low light, black and white light, and the cones produce color for more higher light situations, brighter situations, okay? So these, when they're stimulated, produce graded potentials to stimulate bipolar cells, which is the next one in the line. So those bipolar cells are neurons that are stimulated by the rods and the cones. They send another graded potential to the ganglion cells, and the ganglion cells are where we actually get our first action potential. And those cells are actually the axons of those cells are what make up the cranial nerve 2, which is the optic nerve. So we've got kind of three types of cells there. And here's a close-up of the retina and what that looks like. Okay, so with A there, that's the rods. Okay, B is the cones. Okay, so we can see cones there. C is the next cell down. So these get stimulated first, send that graded potential that direction to the bipolar cells. We get another graded potential here to the ganglion cells, and then those ganglion cells send their signal via an action potential on the optic nerve, which is really just the axons of those ganglion cells. It's cranial nerve 2. Okay, so which one actually sends the action potentials? That's D. That's the ganglion cells. And the interesting part about this is when you think about this, which way is the light coming? Is it coming in this way, which would be 2, or is it coming in this way? Okay, and we said this layer, this pigmented layer of the retina was closer to the choroid. Oops, choroid right here, right? So this would be the back of the eye, right? Excuse me, this would be posterior. This would be anterior, right? So light's got to be coming this direction. So it's 2, right? It's got to be coming this way. So it's kind of interesting that the light passes over the optic nerve, right? Passes over the optic nerve, passes over those ganglion cells, passes over those bipolar cells, and then it hits the cells that are going to be stimulated, the rods and the cones, and then that sends a signal back this way through the bipolar cells, through the ganglion cells, through the optic nerve. So it's kind of an interesting setup there. Okay, so then we got two broader or multiple broader regions of the retina. One important part is called the fovea centralis. Okay, fovea centralis is here. This is pretty much directly behind the pupil. Okay, this is packed with cones. This is where we find most of our cones. This is where high visual acuity happens. This is the focal point. This is where the lens is trying to focus the light. It's the point of focus. Okay, and that's exactly why the optic nerve is offset. Right, we don't want the optic nerve to be right behind the pupil, because right where the optic nerve is a place called the blind spot or the optic disc, because there's actually no photoreceptors right here. Okay, so that's a blind spot. Okay, and then there's the whole periphery out here. All right, so this is where most of our rods are. You can still see stuff in your periphery, but it's not clear, and it's black and white because it's mostly rods. Okay, so what about the different segments of the eyeball itself, and how does the lens fit into this? So the lens is what is the dividing line between anterior and posterior portions of the eye. Okay, so the lens is very elastic. Oops. Pencil stopped working. The lens is very elastic. All right, sorry, had to get my pencil going again. The lens is very elastic, and it's going to bounce back to its original shape unless it's pulled on the outside. Okay, so when that tension is released, it bounces back to its original shape and gets thicker. Okay, so when we want to focus the image with the lens, we have to control how thick the lens gets. Okay, so that changes the degree of light by bending or refracting the light. Okay, the lens also serves as a dividing line between anterior and posterior segments. Okay, so we already talked about the anterior segment having aqueous humor. The posterior segment contains a more jelly-like substance called vitreous humor. Okay, it's also transparent, but because it's more jelly-like, it helps maintain the shape of the eyeball. Okay, very, very slow turnover. So if you rupture your eyeball, like happened here, and you lose some of that vitreous humor, it's very, very hard to maintain the shape of your eye, and your retina actually falls off the back of your eye without the pressure created by the vitreous humor. Okay. All right, so then the anterior segment of your eye, again, contains that aqueous humor, much more fluid-like. It's constantly forming, constantly draining. And there can be some problems with the anterior chamber of the eye if you produce too much fluid. Okay, so it's produced by the ciliary processes, and it's reabsorbed by some sinuses in the sclera. Okay, so the function of it is to nourish the lens and the cornea, right, to provide nutrients because there's no blood supply to that part of the eye. But what can happen with glaucoma is you build up too much pressure in the anterior chamber of the eye. Okay, there's some either a blockage with the drainage system of the aqueous humor, or you're just producing too much aqueous humor. And what happens there is that pushes the lens backwards, and that pressure pushes the vitreous humor backwards, which can smash the retina. It can actually damage the retina and the optic nerve, and that can eventually lead to blindness, and that's called glaucoma. Okay, lastly, we've got these extrinsic eye muscles. Okay, I'm just going to uncover these. One of those boxes was in the wrong spot anyways. This is a review from last semester, right? So there's 4 recti muscles and 2 oblique muscles. Okay, so the recti muscles, superior rectus, inferior medial lateral. Top 3 are all controlled by cranial nerve 3. 4th one by cranial nerve 6. Okay. And you can see the motions that those produce. Okay, this is a review from last semester. Then we have the superior oblique, which does this. So we want to think of that as down and lateral, or down and out. Okay, and then we have the inferior oblique, which does this, up and out. Okay, so upward and lateral. Both of those help with rotation. You can see this one's cranial nerve 4, and this one's back to cranial nerve 3. So you've got 3 different cranial nerves that control these muscles, and it's important for you to know which ones control which. All right, this is just a labeling of those muscles, which you can do here. Okay, which letter is the muscle that moves the eye up and out? Right, so that would be our one from before, the up and out one that we just talked about right here. Right, that's the inferior oblique. So which one is that? We see it. Oops. We see it. This is a superior view, so we're not going to see much of it right there, but we can see it right here, that inferior oblique muscle. Okay. So you want to be able to label those muscles on the you want to be able to label those muscles on the diagram and know what actions they produce. Okay, and this would be a good practice for you to go ahead and walk through this when you're tracking an object upwards, when you're using your eyes to go up. What are you using? You're using your superior rectus muscles on both eyes, right? And then you'd want to know which nerve innervates those. If you're going downwards, using your inferior rectus on both, that's easy. But what about when you track an object to the right? Okay, then you want to think about your right eye. It's going to be the lateral rectus, but the left eye is going medially, right? So the left eye is going to be the medial rectus, okay? And don't worry about involving the obliques. You don't have to worry about that. So an object moving towards you, you're going to use both of your medial recti, away from you, both of your lateral recti, okay? And make sure to include the nerves when you walk through this. All right, and then lastly here, a couple things about the way that we move our eyeballs, okay? The way that we move our eyeballs, our eyes are controlled with very fine motor control, very small motor units, okay? We can move them very small amounts at any given time. There's an idea called saccadic movement, which means that we rapidly jump from one focal point to the next. Think about when you're driving in the car, going through farm country, and you see those rows of crops, right? Your eyes will catch onto one row, so you can actually see down the row. And then once it goes past you, then your eyes skip back to another row, and then you watch that one, skip back to the other row. That's exactly what saccadic movement is, because your eye can't focus if it's just constantly scanning, right? It has to pick a point, focus, and then pick another point, focus. That's what saccadic movement is. But if you're tracking an object that's moving, you can do that, right? So if you're following a car, if you're standing still on the side of the road and you're following a car, then you can do that, okay? But it's different than trying to find a bunch of different things to focus on as your eyes are moving, okay? And I'll go through this one a little bit more on the class for Friday. Okay, so some abnormal movements you can have: strabismus. Strabismus is a medical term for cross-eyed, and diplopia is double vision. And this happens if your eyeballs are looking at two separate things. Okay, if your eyeballs are looking at two things, you get two different images coming in, and that leads to double vision. All right, guys, that's it. We'll see you for the recorded class on Friday.